Welcome to the third lecture on normal physiology. It's about physiology of the autonomic nervous system and humoral control and hypothalamo pituitary system. Objectives which we will discuss today, they are same as the questions which you have in your practical classes. So we will talk about structure and functions of the autonomic nervous system. We will trace the components of the autonomic reflex arc. We will discuss the autonomic ganglia, neurotransmitters, cytoreceptors. We will talk about three main subdivisions of the autonomic nervous system. They are sympathetic, parasympathetic, and metasympathetic nervous system. And we will discuss their role in regulation of functions of visceral organs. We will compare the functions, whether they are synergistic or antagonistic in ANS divisions. We will mention the role of ANS in stress and adaptation. I will give you examples of autonomic reflexes, and we will discuss the hierarchy in ANS regulation of higher bulk structures. We will also talk about structure of the diencephalon with the main focus on the hypothalamic functions. And at the very bottom of the slide, you can see the progress bar, these boxes. Each of them correspond to the questions which we are going to discuss. So this is how you can easily navigate through the lecture if you will be later on watching the video and you will try to forward or go back in slides. So each box will have its number, as you can see here. So that's how you will know which question is exactly on the slide and what we are discussing. Hope it will be helpful. So the first one is about the structure and functions of the autonomic nervous system. As you know, there are three subdivisions: sympathetic, parasympathetic, and enteric nervous system. The first two, they belong to central control. And enteric nervous system, it provides local regulation. Here you can see the centers of sympathetic and parasympathetic nervous system. As you can see, sympathetic neurons, they are located in thoracolumbar area of the spinal cord, while for the parasympathetic, it's the cranial and sacral origin. The main function of the autonomic nervous system is to maintain homeostasis, a rather stable internal environment. So autonomic nervous system, it provides the neural control, that's the nervous mechanism of regulation. And how it does it? Through the regulation of activities of organs, effector organs such as cardiac muscles, smooth muscles, and glands. These are the examples of functions of the autonomic nervous system. So what it does, it helps to maintain the homeostasis in response to normal fluctuations of functions which you control. For example, we have lots of negative feedbacks in the body, like regulation of blood pressure, regulation of heart rate, thermoregulation, all of those. Another one, it's about the integration of stress response, including the response to exercises or classical fight-or-flight reaction. And the last but not least, integration of visceral functions. Because all of the organs in the body, the activity has to be coordinated so that the entire organism will work as one at the optimal level. Now, on this slide, you can see the structure of the reflex arc. So let me remind you from the previous lecture, using the example of somatic reflex arc, the main components of the reflex arc. So this is the somatic. The first one is the afferent, which includes receptor, then goes the sensory neuron, the cell body at the dorsal root ganglion, and then the center, which is the spinal cord in this example. In the anterior horns, you have the second neuron, which is the motor neuron, then goes the efferent fiber and effector organ. So these are the main components of the somatic reflex arc. Here you have two neurons, and this is the example of monosynaptic reflex arc. ANS also operates primarily through reflexes, but in this case it will be about visceral reflexes. So what we have here, you have sympathetic and parasympathetic reflex arc. So how does it work? The afferent pathway, as you can see, it's pretty much similar to the somatic nervous system. You also have receptors, then you have afferent fiber which enters the spinal cord with the sensory neuron in the dorsal root ganglion, but then the next neuron, it will be in lateral horns. This is the autonomic neuron. This is so-called preganglionic neuron. This preganglionic neuron gives rise to preganglionic fibers, which synapse on the next neuron in the ganglion. In case of sympathetic, it might be paravertebral ganglia, which gives rise to postganglionic fibers. In parasympathetic, as you can see, you have rather long preganglionic fibers. Then the neuron is located within the wall of the organ and very short postganglionic fibers. So that's how the autonomic reflex arc looks like. It has 3 neurons if you will count the sensory one, or you can say it has 2 neurons if you are talking only about the efferent pathways. So let's take a look and let's try to compare the sympathetic and parasympathetic reflex arc. You can see the location of neurons will be different. Preganglionic neurons, in case of the sympathetic reflex arc, they are located in thoracolumbar area, while for the parasympathetic it will be either the nuclei of the cranial nerves, you can see cranial nerve number 3, 7, 9 and 10, or if you are talking about the spinal cord, then it will be in sacral segments. Then the next difference, the ganglia where this preganglionic neuron will synapse at. In case of sympathetic, it will be either paravertebral or prevertebral ganglia, while in case of parasympathetic, it's about terminal or intramural ganglia. And as you can see, the length of preganglionic and postganglionic fibers will be different in this case. In sympathetic, you have short preganglionic and rather long postganglionic. In parasympathetic, it's rather long preganglionic and rather short postganglionic fibers. The next difference, it's about the neurotransmitters which will be released in these synapses. If we are talking about a preganglionic neuron, in both cases you have acetylcholine as a neurotransmitter and you have nicotinic cholinergic receptors. It doesn't really matter if it's sympathetic or parasympathetic nervous system. But in postganglionic neurons, there will be a difference. For example, in case of sympathetic nervous system, it's norepinephrine which is secreted. And in case of parasympathetic, it's acetylcholine. And receptors on effector organs also will be different. Like in case of norepinephrine, these are the adrenergic receptors, alpha and beta. In case of acetylcholine in parasympathetic, it's M cholinergic receptors. And also what you have here, this is the type B thin myelinated fiber, preganglionic. And it is also cholinergic in both cases, no matter if it's parasympathetic or sympathetic. For postganglionic, it's type C, thin, unmyelinated, but here it will be cholinergic and here it's adrenergic. So these are the main differences. And here are the slide once again about the comparison of autonomic reflex arc and somatic, with the main focus on efferent part. As you can see, the cell body of the motor neuron for the somatic one is at the anterior horns, while here it's in the middle in the lateral horns. Then there is no neuron outside the spinal cord, while for the ANS you do have one at the ganglia. So at least these few differences, and also you have a small summary in this table which describes the differences between somatic motor and autonomic motor branches of the reflex arc. Now let's talk about autonomic ganglia and neurotransmitters. This is the sympathetic reflex arc, and here you can nicely see the location of cell bodies of postganglionic neurons. So the first type of the ganglia which we can have is paravertebral ganglia. These lies on both sides along the spinal cord, and they will form the sympathetic chain or sympathetic trunk. But some fibers, as you can see, they pass through paravertebral ganglia, and then they make a synapse in prevertebral ganglia, which are a little bit further away from the spinal cord, and then you have long postganglionic fibers. This is what we have in sympathetic nervous system. In parasympathetic nervous system, you remember we have very long preganglionic fibers and rather short postganglionic ones, because the ganglia which we have, they're very close to the effector organ, and they will be either close to the organ, which is called terminal ganglia, or even within the wall of the organ, which is called intramural ganglia. Here once again you can see the neurotransmitters and receptors which we have in pre and postganglionic neurons for both sympathetic and parasympathetic, with the location of centers. And there is one exception about innervation of the suprarenal gland, its medulla. You can see there will be preganglionic neuron which is cholinergic, which gives innervation to enterochromaffin cells, which will then release epinephrine and norepinephrine from the medulla of the suprarenal gland. But as you can see, there are no postganglionic fibers. So the medulla of the suprarenal, you can say it's the ganglia, kind of a ganglia itself. And here you can see the ratio of epinephrine and norepinephrine, which are secreted from the medulla. Now once again you can have a look at the ganglia which we have. Here this diagram nicely shows you the exact locations. So these are the paravertebral ganglia or sympathetic chain. These are the prevertebral, you can see superior, inferior mesenteric ganglia, celiac ganglia. This is for the sympathetic. And for the parasympathetic, you can see they will be within the organ. So you have very long preganglionic fibers and rather short postganglionic fibers, because all the ganglia, they are either close to the organ or within the organ. Now let's talk about cytoreceptors. Since we have two neurotransmitters, we will have two types of receptors. The first one, adrenergic receptors. They are located at organs which are innervated by the sympathetic nervous system. And as you can see, there is a slight difference in affinity. Like norepinephrine, it binds mostly to alpha adrenergic receptors, while epinephrine, it can bind to both alpha and beta receptors. Why is it important to know? Because later on, when we will study the cardiovascular system, you will see that effects which are provided by these substances, they might differ, and it will be determined by the type of the receptor they act on. So as you can see, for example, in this case, if the hormone binds to alpha adrenergic receptors, you can have vasoconstriction, and this is what you have in blood vessels. Or if it's about the heart, here you have beta adrenergic receptors, and it increases the heart rate, which is called tachycardia. The second group of receptors, it's for the acetylcholine. They are called cholinergic receptors. They are located at parasympathetic effector organs, so those organs innervated by the parasympathetic nervous system, and also few organs which are innervated by the sympathetic nervous system, and that's the exception, like the sweat glands, because there the neurotransmitter is also the ACH. So that's for postganglionic. And you remember that all preganglionic neurons, they all have acetylcholine as a neurotransmitter. And from excitable tissues, I hope you remember that muscle cells of the somatic nervous system, in the neuromuscular junction, the neurotransmitter is also the acetylcholine. So types of receptors which we have, they are muscarinic and nicotinic. And for muscarinic, they are further subdivided into types like M1, which are present in the GI tract and CNS, M2, which are present in the heart, M3, which are present in smooth muscles. And for nicotinic receptors, there are also at least two subtypes which you should know. One is nicotinic neuronal, that's between pre- and postganglionic neuron, and nicotinic muscular, that's at the neuromuscular junction. So here you can see the example of how it interacts with the effector organ. So here you have nicotinic receptors, because that's the preganglionic and postganglionic neuron. So here it's nicotinic receptors in the ganglia. But when it comes to the effector organ, here you have muscarinic receptors like M1, M2, M3. And in case of the heart, acetylcholine will cause decreased heart rate, which is called bradycardia. So let's talk about sympathetic nervous system. Sympathetic nervous system, it's responsible for fight or flight response. This is when the person is in danger or if there is a stressful situation. How sympathetic nervous system can help you in such case? It can act directly on effector organs and it can boost their activities, or it can act through sympathoadrenal system, sympathetic preganglionic fibers, remember, which innervate the adrenal medulla, which leads to massive secretion of catecholamines, epinephrine and norepinephrine. So here you have a summary on what happens during fight or flight response. Why is it called fight or flight response? Because if you are in danger, you can either fight, or if you are not going to fight, you can run away, which is flying, in other words. So how sympathetic nervous system can be helpful in such case? As you can see on the slide, critical activities which are essential for survival, they will be upregulated. So what does that mean? Like pupils will dilate for the better visual control of the situation. Bronchioles will dilate because you have relaxation of smooth muscles, as well as respiratory rate will increase to provide you more oxygen. Then cardiovascular system also will be activated. The heart rate increases, the stroke volume rises, the blood pressure increase. For the same reason, to provide better oxygenation and more nutritive substances to all organs all over the body, so that you can fight with the stress. And epinephrine also, it causes breakdown of the glycogen, which will rise the glucose level in blood, because you need energy during stressful situation. And those non-critical activities for survival, they will be downregulated. And one of the examples of such systems, that's the gastrointestinal system. Definitely you do not need digestive system if you are in danger or stressful situation. So here also you have a small summary over the organs which will be activated or which activity will be suppressed. Now the third question, it's about the parasympathetic nervous system. As we already mentioned, we have cranial region. So here you can see the nuclei of the cranial nerves like oculomotor, then the facial, then the glossopharyngeal, and vagus. That's the main parasympathetic nerve, which gives innervation to most of the organs in the body. So this is the cranial origin. And also we have centers in the spinal cord. These are the sacral segments. Parasympathetic nervous system could be described as rest and digest system. So what happens in this case? It will be just the opposite. Like critical activities for survival, they will be downregulated, and non-critical activities, they will be upregulated. So basically here you have a comparison of what happens in case of parasympathetic activation. You will have constriction of pupil, stimulation of flow of saliva, because remember, you call it rest and digest. and definitely saliva is needed for the proper digestion to occur. And here you can see stimulation of digestion as well. Not only secretory activities, but also the peristalsis of the digestive organs. And another case when it's activated during rest. And as you know, at rest, all of the activities will be slowed down, like the heart rate decreases, the respiratory rate decreases, metabolic processes, they slow down. So this is the parasympathetic nervous system. The next one is the enteric nervous system. Sometimes it's being called little brain in the gut. And if you will look at it, it's really so, because here you can see the receptors, they are within the wall of the intestine. Afferent fibers, the cell bodies of neurons, efferent fibers, and the effector itself, everything is within the wall. So the entire kind of reflex arc is still within the wall. And what are the functions? You have submucous plexus, which mostly stimulates secretion from the mucosal epithelium. And as for the myenteric plexus, auerbach's, mostly it will activate the intestinal motility by stimulating the peristalsis, and it inhibits contraction of the sphincter, which means sphincters will relax so that the food can move out through the digestive tract. Now, let's talk about antagonism and synergism, whether sympathetic and parasympathetic, they're totally opposing each other, or maybe there are some other options as well. So here you can have a summary of main effects of sympathetic and parasympathetic nervous system on different organs. So this is what we have mentioned already. Also in this summary you can find receptors which these neurotransmitters act on to provide definite effect. Now, as you know, most of the visceral organs, they receive dual innervation, and in most of the cases actions of sympathetic and parasympathetic divisions will be opposite to each other. Like for example, you can see for the heart, sympathetic increases the heart rate, contractility, while the parasympathetic decreases the heart rate and decreases the contractility. But it's not always the case. Some organs, there are some exceptions. We have organs without dual innervation, like adrenal medulla. It will be only innervated by the sympathetic nervous system. Then erector pili muscles in the skin. The sweat glands in the skin, they do not receive parasympathetic innervation. And most of the blood vessels, they also have only sympathetic innervation. So how do we control the functions of such organs? In these cases, regulation will be achieved by increased or decreased activity, that's called tone or firing rate, of the sympathetic fibers. Let's say if you want to have constriction of cutaneous blood vessels, you need to increase sympathetic activity, means more impulses should come. And these will stimulate alpha-adrenergic receptors. And if you want vasodilation to occur, then you need to decrease the sympathetic nerve stimulation. So that's how activity will change. And this is what you have in this table. And that's why here nothing is written, because there is no parasympathetic innervation of these vessels. Now, other options of interaction, they include complementary and cooperative. So let's talk about cooperative action. It's about the reproductive system. We have two processes. Let's talk about the male one. We have erection of the penis, for example, that happens due to the vasodilation, which result from parasympathetic activation. And the next step is ejaculation, is due to the stimulation throughout sympathetic nerves. Even though these two processes, they are not opposite, but they do cooperate together to enable a proper sexual function in male. Then, example of complementary activity it could be described using saliva secretion as an example. So what do we have in this case? As you can see, parasympathetic causes secretion of watery saliva, while sympathetic one produces rather viscous, mucin-rich saliva. So how does this happen? And why do they complement each other? Parasympathetic produces watery saliva, while sympathetic nerves, they will cause constriction of blood vessels throughout the digestive tract, and it will lead to decrease in blood flow, and salivary gland will produce a thicker and more viscous saliva. So that's how they work in a complementary fashion. Now let's talk about role of ANS and stress and adaptation. We partially mentioned that. Mostly it will be governed by sympathetic division. This is when you need an urgent, a quick response. But as you remember, we have two mechanisms of regulations. One is neural and one is humoral. So in case of a stressful event, which will be received through the hypothalamus, there will be two pathways activated. One through sympathetic neurons, which will go to the adrenal medulla, which is the sympathoadrenal nervous system, which will stimulate production of epinephrine and norepinephrine, and it will lead to short-term responses. This is the left one. Short-term responses such as increase in heart rate, increase in blood pressure, bronchodilation, increase in glucose level in blood. So all of those adaptive responses of the body which will help you to survive. But on another hand, later you will learn that hypothalamus through anterior pituitary can stimulate the adrenal cortex, which will give you one more hormone, which is the cortisol. That's what is called stress hormone. And cortisol, that's the glucocorticoid, it will produce long-term responses because humoral regulation, it's slower, it takes time, and it's rather long-lasting. And these are the positive effects of glucocorticoids, cortisol, during the stressful event. That's what you need for a better adaptation. So why did I use this picture at the very beginning for the title of the lecture? There is one thing you should understand, that sympathetic and parasympathetic nervous system, they are not fighting with each other, even though most of the functions are opposite. Sympathetic and parasympathetic nervous system, they're kind of balanced. Each of them, one of them, will be activated only in case of need. Like if there is a stressful event, sympathetic gets activated. As soon as the stress is gone, it goes back to its initial level to be back balanced again. While parasympathetic gets activated if you eat some food and digestion is initiated. But as soon as digestion is over, parasympathetic is returned to its initial condition. So these two should be balanced. They are not fighting, but they are working together to maintain homeostasis. That's what you should understand about these two branches of the ANS. Now, as for the autonomic reflexes, there are three types, and that's what you're going to do during your practical classes. Visceral, these are the reflexes which are when one organ gets stimulated, the activity of another organ might change. During your practical classes, you will examine ocular cardiac reflex. This is when pressing the eyeballs, it will slow down the heart rate. And here you can see the reflex pathways for this reflex. Now, viscerodermal, if there are some pathological processes in inner organs, we have certain projection areas on the skin. This is so-called referred pain, which can be used as a symptom of that organ damage. The one you might be familiar with, it's about the heart, like myocardial infarction. When there is diminished blood supply to the heart, then myocardial infarction might develop. But in this case, the person might not complain on pain in the heart region itself, but rather the person might be complaining on pain in his or her left hand or left arm. And that's your responsibility to recognize that this increased sensitivity or this pain sensation could be a signal of heart cardiac problem. And the third one is dermatovisceral. This is when you use certain manipulations and you apply them on the skin, and it will change the blood supply of underlying organs. Like some physiotherapeutic procedures are based on these reflexes. Now the next question number 7, it's about central regulation of the autonomic nervous system. So as you know, in nervous system we have a hierarchy relationship. So you have the higher up above centers which control the below one. So same does for the autonomic nervous system. Mostly it will be about limbic system. So we have suprasegmental, which is the limbic. Also we have subcortical centers and hypothalamus. That's the most important structure for the autonomic control. These structures, and also we have few centers in the brainstem, partially reticular formation contributes to them. So these higher centers, they also play important role in motivation, in behavior, in emotions, and those reactions are also driven by the hypothalamus and the limbic system. And before, in previous lecture, we discussed that in brainstem we have nuclei which control basic homeostatic functions, such as we have cardiovascular, respiratory, digestive centers throughout the brainstem. And all these central structures, they send the influences down below to the spinal cord, and this is where we have the lower motor neuron of the autonomic nervous system, the efferent neurons, which then will send efferent fibers to effector organs. So this is how the regulation happens. Now, the structure and functions of the limbic system, the last question within this chapter. Limbic system, the nuclei of the limbic system, as you can see in the scheme, they are divided into archaeocortical, paleocortical, juxtacortical, and subcortical. And here you can have the examples of structures which are included into this. Main functions of the limbic system, it's regulation of emotions, behavior, including feeding behavior, regulation of sexual drive and sexual behavior, goal-directed behaviors. Limbic system also regulates the functions of the visceral organs because of the hypothalamus, which is one of the structures. And these are the components of the limbic system, which consists of a group of 4 brain nuclei and fibrous tracts, which will form a ring around the brainstem. So these are the components. Now, the main focus out of all structures will be on the hypothalamus, because as I said before, hypothalamus plays very important role in regulation of the autonomic nervous system. And as you might know, the hypothalamic nuclei, they are divided into several groups. So we have anterior group, posterior group of nuclei, lateral and tuberal group of nuclei. And here you have few examples of centers of the located within the hypothalamus. Like we have centers for hunger and satiety, which regulates the feeding behavior. Then we have centers for thirst and water regulation and electrolyte balance. We also have centers for the heat loss and heat gain. We also have centers which control sexual behavior, circadian rhythms, and also we have centers for the endocrine system, for the releasing factors, and for the ANS as well. Sympathetic and parasympathetic nervous system control, it comes from the hypothalamus. Why hypothalamus can do that? Because it receives various afferent and efferent inputs and connections. So as you can see, it receives it from visceral and somatic sensory pathways to integrate regulation and homeostatic control. Then also it receives afferents from the brain cortex, like frontal lobe, and from the limbic system, and sends it to higher centers to contribute to mood and emotion control. Then, as a part of efferent connections, it will be connected to pituitary gland, through hypothalamo-hypophyseal tract or to the posterior pituitary, that's the posterior pituitary, or through the portal system to the anterior pituitary. Then also efferent neurons, they pass to the brainstem nuclei to influence the parasympathetic outflow, or also it goes to the lateral horns of the spinal cord for the sympathetic outflow. And it is also connected to the reticular formation and limbic system. So here you have once again a diagram which shows the connections between hypothalamus to the ANS. Either brainstem, the cranial part of the parasympathetic, or spinal cord, which is the sympathetic and parasympathetic connections. This is how hypothalamus contributes to the autonomic control. But beside that, as we have mentioned, we have hypothalamo-pituitary system. This is where hypothalamus produces releasing hormones for the anterior pituitary, which will produce tropic hormones to act on various endocrine glands. So as you can see, functions of the autonomic nervous system, they are highly integrated with the endocrine regulation, because hypothalamus, it controls both efferent autonomic nervous system activity and it influences secretion of the hormones by the through the pituitary gland. So as such, hypothalamus, it controls most of the homeostatic functions either through ANS or through the endocrine system, because it has various output pathways, and that plays a huge role in maintenance of homeostasis and regulation of functions of visceral organs. So now the second part of our lecture will be about humoral control. And here you have the new set of objectives and questions which we are going to discuss. And as you can see, the new progress bar comes, which means we still have to discuss 7 more questions. So here it will be about humoral control of body functions. We will revise once again the comparison between the humoral and neural control of body functions. I'll give you classification of humoral factors. We will talk about structure and functions of endocrine system, about hormones, their classification properties. We will discuss how hormones, they act on target cells. We will talk about different receptors, secondary messengers. We will mention the hypothalamo-hypophyseal system and the functions of the hormones which are produced with their effects. So the first question which we have, it's about mechanisms of regulation. So remember, we have two. One is humoral and one is neural. And to refresh the differences, humoral regulation happens through biologically active substances, while nervous through impulses. Then humoral, it's rather slow and long-lasting, while neural is fast and short-lasting. Here you have a diffuse, generalized response, while the neural control is very well localized and it has an exact address, because it's how the innervation of organs happens. Now, why the regulation is called humoral? Because humor means liquid. So various factors which you have in bodily fluids, they're responsible for the regulation. Examples include like ions. Remember, in excitable tissues you used to have calcium ions, which helped in synaptic transmission, for example. Then you have different metabolites like lactic acid, pyruvates, and so on. Utilizones, the waste products which are produced like hydrogen ions, because you know during activities pH changes, and CO2 levels. Hormones, which could be divided into classic hormones and tissue hormones. And neurotransmitters. So that's the classification of humoral factors. Now let's talk about the endocrine system itself. So as you know, endocrine glands, they lack the ducts and they secrete their products, which are the biologically active substances, hormones, directly into the bloodstream. So you can see the endocrine cell, it releases hormone into the bloodstream and then it travels through the blood to reach the target cell. Why is it called target cell? Because it has receptors on its surface for this particular hormone. And target cell, which has these receptors, Then after forming the receptor hormone complex, it will change its function within the cell. And of course we do have the mechanism which then turns off the action of this hormone. Now, the endocrine system consists of glands which could be divided into central and peripheral glands. We also have temporary glands like thymus and placenta. We also have mixed glands like gonads and pancreas. And we do have organs with endocrine activity. They are not glands, but they can still produce hormones such as like kidney, heart. So let's take a look at the central glands. We have hypothalamo hypophyseal system, as we have mentioned before. So what we have here, we have hypothalamus, which is connected to anterior and posterior pituitary, either through portal system or tracts, to produce tropic hormones which act on most of the peripheral glands to cause their secretions. So let's talk about classification of hormones. That's the third question. The first one is chemical classification. Here they are divided into amines, polypeptides and proteins, and steroids. Amines derived from amino acid, tyrosine and tryptophan. These are the hormones of the adrenal medulla, thyroid gland. Polypeptides and proteins, examples are antidiuretic hormone, insulin, and steroids. These are the sex hormones and glucocorticoids. The next one, it's about synergists, antagonists, and those hormones which have permissive effects. So for antagonism, it's easy, those hormones which have opposite effects like insulin and glucagon. Hormones with permissive effect, it enhances the responsiveness of a target organ to a second hormone or increases its activity. Like the example which we can use, estrogen estradiol has a permissive effect on the responsiveness of uterus to progesterone during the pregnancy, for example. Synergistic action. Here you have epinephrine and norepinephrine on the heart. If they act together in same concentrations, they stimulate or they show even greater increase in cardiac rate. Now, another classification based on how far hormone has to travel from the place it was secreted. If it doesn't travel far but rather acts on its own cell, like the cell produces, and then hormone acts on that cell. This is what is called autocrine signaling or autocrine effect. Now, the next one is paracrine. Hormone is secreted, but it doesn't travel too far. It acts on adjacent target cells. This is what is example which can be used as APUD system. But in most of the cases, hormone, once it's produced, it travels far from the place it was produced, and it acts on target cells which could be located very far from the secretion itself. Now, the next one is biological regulation. Here you have morphogenetic action. These are the hormones which are responsible for growth and development. Metabolic, these are the hormones like, for example, insulin and glucagon, which controls metabolic processes. Then goes kinetic. In this case, hormones, they initiate or trigger the activity of the organ. Then we have under biological, corrective, which is the adjustment of the function of the organ. And the last one is the functional one. Here hormones could be divided into releasing, tropic, effector. Releasing, they are produced by hypothalamus, tropic anterior pituitary, and effector hormones by peripheral glands. Now, all of the hormones, they have certain properties. That's how you can describe their activity. As we have mentioned, most of the hormones, they have distant action. So they act quite far from the place they were produced. These are the highly active substances, which means even minute concentrations will produce the effect. Then they have specificity of action, which means insulin will not increase heart rate or will not cause water reabsorption, but the function of insulin is to control the glucose level. That's the specificity of action. The next one is velocity of production. For some hormones it takes seconds, and for some it will be about days. And the last one, duration of action. For how long the duration of action will last. Here also there are different options for different hormones. Now, as we already mentioned, hormones will be released by the endocrine gland directly into the bloodstream, and then they have to reach the target cell. And for interact with the cell, you need receptors. Here we have several options, and it depends on the chemical nature of the hormone, whether it can enter the cell or it cannot enter the cell. So how? Steroid hormones, as you know, they are lipophilic, and the cell membrane of cells, like of target cell, it's made about bilipid of phospholipids, biphospholipid layer. So in this case, steroid hormone, it can go through the cell membrane and it can enter the cell. So for steroid hormones, receptors will be located within the cell or within the nucleus, because steroid hormones, they can enter the cell. So two types of receptors, cytoplasmic or nuclear receptors. So once it reaches the receptor, then it binds to it. You will have hormone receptor complex. Then what happens next? There will be DNA transcription, which gives you new mRNA, which then goes into translation and gives you the new protein synthesis. And these newly synthesized proteins, they will produce various physiological actions. Now, what about non-steroid hormones, proteins and peptides? These hormones, they cannot enter into the target cell. So for them, receptors will be located in the cytoplasm, in the cell membrane. So here, hormone binds to the receptor, which is in the cell membrane, and there will be stimulation or inhibition of the G protein, which is attached on the inside of this receptor, and changes in activity of this G protein activates secondary messenger. So this is basically the messenger which will produce or which will trigger the effects inside the cell. So in this case, you have example of CMP as a secondary messenger. So what we have, activation of adenylate cyclase, which converts ATP into CMP. Then CMP further activate the protein kinase, which will leads to phosphorylation of proteins and produces various physiological actions. Besides CMP, you can have cGMP, IP3 inositol triphosphate, and you can also have calcium ions. So these are the mechanisms of hormonal action on the target cell. The next question, which we will talk about, it's about hypothalamo hypophyseal system. So here on this diagram, you have two, anterior and posterior pituitary. So for hypothalamo pituitary connections, we have two separate systems. One connects the hypothalamus to the anterior pituitary, and another one connects hypothalamus to the posterior pituitary. So how does these connections occur? For the anterior lobe of the pituitary gland, it is linked to the hypothalamus via the portal system, blood vessels. So here, the hypothalamus, the nuclei of the hypothalamus, they synthesize releasing factors. We have releasing and inhibiting hormones. These hormones will be delivered through the portal system capillaries into the anterior pituitary, and at the anterior pituitary they will stimulate production of the tropic hormones. Like here you have example with the growth hormone. We have growth hormone releasing hormone and we have growth hormone inhibiting hormone. One stimulates and one inhibits production of growth hormone itself from the anterior pituitary. And for the posterior pituitary, what we have in hypothalamus, there are two nuclei, supraoptic and paraventricular nuclei, which will synthesize two hormones, oxytocin and antidiuretic hormone. These hormones synthesized in these neurons of these nuclei through the axons of these neurons will be delivered to the posterior pituitary, where they will be released into the bloodstream. So that's the connection of hypothalamus to the posterior pituitary. Now, let's have a look at the releasing hormones. What do we have in here? So in this table, you can see a summary of the releasing hormones that we have in hypothalamus for the anterior pituitary. Like we have growth hormone releasing hormone and somatostatin, which will stimulate or inhibit production of the growth hormone that will cause growth and development of organs and tissues. Then we have thyrotropin releasing hormone, which initiates the production of TSH, thyroid stimulating hormone, by the anterior pituitary, and this in turn will act on the thyroid gland to produce thyroxine and triiodothyronine. Then we have corticotropin releasing hormone, CRH, which will lead to the production of ACTH, that's the adrenocorticotropin, which acts on the adrenal cortex to produce cortisol. Then we also have gonadotropin releasing hormone, GnRH, which will stimulate the production of follicular stimulating hormone and luteinizing hormone. And for prolactin, we'll have dopamine, that's for the mammary gland and milk production. So that's the connections of the hypothalamus and anterior pituitary and hypothalamo-hypophyseal system. So actually all of the glands, they could be divided into two types, either pituitary dependent or pituitary independent. Pituitary dependent glands, they are on the hypothalamo-hypophyseal system. So what we have here, hypothalamus will produce releasing hormones which will act on anterior pituitary, which will produce pituitary or tropic hormones, which will act on peripheral endocrine glands, and they will produce the effector hormones, which will produce some physiological effects. This is true for some of the glands, but some glands will be pituitary independent, like parathyroid gland or pancreas, which will not be regulated by the pituitary or hypothalamus. And this is all about the hypothalamus and anterior pituitary. Here you have a summary of the feedback control over the target gland hormones. If their level decreases, then the feedback will be sent to the hypothalamus and anterior pituitary to initiate production of releasing hormones and then tropic hormones to bring the hormonal level back to the normal. If hormones levels will be too high, then inhibitory feedback will be sent to suppress production of releasing hormones and tropic hormones, again to normalize the level of the hormone in the blood. So let's talk about hormones of the anterior pituitary. As we already mentioned, we have TSH, thyroid stimulating hormone, which act on the thyroid gland to produce T3, T4. Then we also have gonadotropic hormones, LH and FSH. The function of them, you have follicular stimulating hormone, which promote the ovarian follicle development and estrogen secretion in females and spermatogenesis in males. Luteinizing hormone, it promotes progesterone release from the corpus luteum in females, and in males you will have androgen secretion. Then we have ACTH, adrenocorticotropic hormone, which acts on the suprarenal gland cortex for the cortisol production, prolactin for the breast development and milk production, and we also have growth hormone which might act on organs and tissues through insulin-like growth factor for growth and differentiation. And these hormones will be produced by the cells, somatotrophs, lactotrophs, gonadotrophs, tyrotrophs, and corticotrophs respectively. And here is the summary on functions and hormones secreted by the anterior pituitary. Now, as for the hormones of the posterior pituitary, what do we have here? Two hormones will be produced in the hypothalamus. They are the antidiuretic hormone and oxytocin. These two hormones are synthesized in the hypothalamic nuclei, supraoptic and paraventricular, and they will be packed into the secretory granules. These granules will travel down along the nerve axons of those neurons to the posterior pituitary, and that's where they will be stored until it's necessary to release them. So hormones of the posterior pituitary, they are not produced in the pituitary gland itself, but they are produced in the hypothalamus and stored at the posterior pituitary. So let's talk about functions. For the antidiuretic hormone, it comes primarily, mostly from supraoptic nuclei of the hypothalamus, and it's responsible for the regulation of plasma osmolarity. How osmolarity could be regulated? By increasing permeability of water in lead distal convoluted tubules and collecting ducts. So antidiuretic hormone, as it comes from the name, diuresis means amount of urine which is produced. Antidiuretic means it reduces the amount of urine which is excreted. So antidiuretic hormone, the primary function is to initiate water reabsorption by increasing the permeability of tubules in the kidney. And it also has one more function, constriction of blood vessels. That's why sometimes this hormone is also called vasopressin. And those people who lack antidiuretic hormone, they might develop diabetes insipidus. This is the disease when abnormal amounts of urine will be produced. This person can urinate as much as up to 20-30 liters per day. And excessive urination, when lots of urine is produced, this symptom is called polyuria. Poly means a lot, lots of urine produced. Second hormone, which is produced by the hypothalamus and secreted by the posterior pituitary, is the oxytocin. This one comes from paraventricular nuclei of the hypothalamus, and it mainly causes contractions of the uterus during the parturition. As well, it also acts on myoepithelial cells in the mammary gland, and it causes ejection of the milk from the breast when stimulated by suckling. So here you have feedback control for the oxytocin secretion. And here you can see during labor, uterine contractions, they force the baby head into the cervix. These will stimulate the stretch receptors in the uterine cervix. That's how the stimuli will be detected. Then through nerve impulses, it will be sent to the hypothalamus posterior pituitary. That's the control center, which will release the oxytocin, which will act on uterine smooth muscles to produce stronger contraction force on the baby head and entire body to push it further into the cervix, which will in turn, as you can see through the feedback mechanism, will further increase the production of oxytocin, and contractions will become even stronger. So this is the example of positive feedback, when you have amplification of the effect. The stronger contraction becomes, the more oxytocin is produced, the stronger contraction will be, and it will keep on going like that until the baby will be born. So that's the only thing which can stop this loop from happening. Now, one more question which we have to discuss, it's about hormones of the pineal gland. Pineal gland is also called the biological clock of the body because it's responsible for the regulation of the circadian rhythms or 24-hour cycle of different functions within the body or secretion of the hormones which we are interested in today. So how does this happen? We do have suprachiasmatic nucleus, which is responsible for the synchronization of bodily functions, and it happens through the endocrine axis, where you have the pineal gland as one of the glands. So how this occurs? You have suprachiasmatic nuclei, which communicates to the pineal gland through the neuronal connections, and also to the brainstem and spinal cord through the sympathetic connections. So what happens? Pineal gland might or might not secrete melatonin directly into the circulation, and melatonin will synchronize all the bodily processes. Like here you can see secretion of cortisol, production of thyroid hormones, activity of cardiovascular system, some metabolic effects. So how secretion of melatonin is being regulated? If there will be light coming to the retina during the day, the sympathetic pathways from the suprachiasmatic nuclei to the pineal gland, they will be activated, and melatonin synthesis and its secretion will fall until the dark. So during the night, when no light comes to the retina, there is no activation of sympathetic pathways. So melatonin production will keep on going. So actually it's alternating production of melatonin and serotonin, which is quite sensitive to changes in length of the day, seasonal changes, or the amount of sleep which you might get. So pineal gland, the circadian rhythms and biological clock of the body. And I'm going to give you one of the examples of feedback control for one of the hormones of the anterior pituitary. That's the growth hormone. So growth hormone, it's secreted by the anterior pituitary, and as we have mentioned, we do have growth hormone releasing hormone and growth hormone inhibiting hormone, or somatostatin. So how it works? If there is increased concentration of insulin-like growth factor or increased concentration of growth hormone itself, the feedback will be sent to both anterior pituitary and hypothalamus, and since it's a lot, then growth hormone releasing hormone will no longer be produced, but rather you will have somatostatin, which suppresses the further production until the levels of growth hormone will become back normal. But if there is too little of insulin-like growth factors or too little of growth hormone, then feedback will be sent to hypothalamus and anterior pituitary to increase production of growth hormone releasing hormone, for the anterior pituitary to get activated to produce you more of growth hormone. So this is the example of negative feedback and how it works. And here on this slide you also can see some of the effects of the growth hormone on peripheral tissues. Because it's responsible for growth and development, that's the process which requires lots of energy, then it increases glycogen breakdown to increase the blood glucose level through the liver. Then also its growth effects will be responsible for the linear growth of the bones and increase in muscular mass. Then also it's responsible for cellular growth and proliferation, and also it causes lipolysis and decreases glucose uptake by the peripheral tissues. And here you have factors which will stimulate or inhibit growth hormone secretion by the hypothalamo pituitary system. Thank you for your attention and have a nice day.